Good morning or good afternoon to everyone. So thank you for joining me. Today, as I say, I'm going to talk about a model for what we call chase and run interactions. And this is going to be divided into about 4 sections, basically. The first, I'm just going to talk about the general framework that we use to model these processes. That's what we call non-local advection diffusion models. I'm then going to talk about a kind of a previous application of these models to the problem of cell sorting by differential adhesion. Then I'm going to kind of move on to talk about what I mean by chase and run interactions before talking about the main part of this, which is really what we call collective chase and run behavior. So the type of things that I'm interested in modeling are what I guess we would call interaction-induced movement. So this is a kind of a ubiquitous process that kind of happens across a whole range of biological scales and is where the interaction between individuals induces some sort of movement response. So a classic example of this occurs in migrating cells through the process of contact inhibition of locomotion. So these images here show 2 migrating neural crest cells that are at the start of this video, at the start of these sort of timeframes moving towards each other. And then they contact each other at a certain point. And that induces then a repolarization and a reversal of their movement direction. So this is an example of an interaction-induced movement between cells where their contact results in them changing direction and moving in another way. And then this is also, of course, something that happens across in animal populations as well, and can even occur at very large scales, is the case of baleen whale populations, which of course use sound and songs as a way to communicate. So, and because of the propagation of sound in the water, those sounds can travel tens to 100 kilometers and induce other whales to move in response to them. So these interaction-induced movements occur across a very wide range of biological populations. The framework that we've been using to model these are kind of broadly kind of belong to what we would call non-local advection diffusion models. And these have kind of got quite a long history in mathematical biology, sort of extending back over about 50 years or so. And they've been used in various applications. And they basically have the following form. They're kind of a non-local partial differential equation for where if U is my kind of population density, which could be either cells or animals, the key non-local interaction occurs in this non-local advection term, where that's effectively describing how non-local interactions with other individuals induce a movement of the population. So the key sort of terms that enter into this sort of model is this kind of interaction kernel, this K of xi, which defines the direction and the dependence on the interaction distance. A function f of u that describes how the population density how the kind of size of that influences this process. The key parameters that I'll particularly be talking about are what we call the interaction strength parameter, where the sign of that reflects what type of response you get. And so here, alpha greater than 0 would induce an attraction response, alpha less than 0, a repulsion response. And then xi is the interaction range parameter, which effectively describes the range over which these interactions are occurring. And F, finally, of course, describes the standard population growth. So these kind of models, as I say, have got quite a long history in mathematical biology. I think the very first sort of ones that I know of were sort of proposed almost 50 years ago. And they've been kind of almost sort of like rediscovered or kind of reformulated ever since. And that's kind of reflects the fact that these models have been used to describe a lot of different sort of processes. So in their original application, they were very much motivated by problems of animal swarming, such as sort of locust swarms. That was in the sort of late 70s, early 80s. From around the 2000s, they've been used to model cell movement, adhesion, 2010s, territoriality, and much more, and more recently, actually, in problems to do with things like traffic flow and opinion dynamics. And if anybody's kind of interested in these sort of models, together with Thomas Hillen and Jonathan Potts, I wrote a review article last year that's describing both these, the kind of the mathematics of these models and also some of the various sort of applications that are being used. So that's kind of the general framework. And to kind of first illustrate their kind of utility, I'm going to talk a little bit about the classic problem, which is cell sorting via differential adhesion. So cell sorting is a phenomenon that you can see in various heterogeneous mixtures of cells. And basically, the classic sort of experiments here is you kind of take two cell types from a kind of a tissue. You can see the kind of, you know, there's kind of this either black, more kind of darker cells and kind of grayer cells here, basically. And you take them out of their tissue, their normal tissue configuration, and mix them up into a kind of a sort of randomized mixture. And then over time, they resort back into the configuration that they have normally found. In this case here, those black cells become encapsulated by the grayer cells, basically. So this is a kind of classic experiments on this kind of date back to, I think, around the 1940s or so, and it's been intensively studied. And it's believed to be absolutely fundamental for correct tissue organization, maintenance and development. But also it's been used as a way to, as a validation, a test for models to describe cell adhesion type behavior, which is, of course, a absolutely fundamental property of cells that allows our tissues to remain intact. So one of the most sort of famous theories to describe these sort of cell sorting sort of processes is that of Steinberg in the 1950s, and it's called the differential adhesion hypothesis, which basically postulates that if you have a kind of two cell types called, say, blue and red cells, then you can effectively define different sort of adhesion strengths between these cells, the fact that these cells kind of attach to each other and adhere with some strength. So that kind of gives you three parameters for the strength between blue and blue cells, the strength between red and red cells, and the cross adhesion strength between the red and blue cells. And then in the differential adhesion hypothesis, it basically predicts that you will get a range of different sort of sorting type behavior from a kind of a complete sorting of those two populations into two separate groups, to a partial sorting type behavior, to that encapsulation that we saw in the previous picture, to a completely heterogeneous mixing of these populations. And the different types of pattern are driven by the precise configuration of these cell adhesion parameters. So, as I say, this was a kind of a model proposed to describe these sort of cell sorting behavior based on distinct adhesions between the different cell types in the population. So that's our kind of testing bed to kind of look at the effectiveness of these non-local models, and you know, probably one of the more successful types of continuous model to describe this cell sorting behavior is through this kind of non-local formulation. And this was actually a problem that I, together with Jonathan Sherritt and Nicola Armstrong, first worked on about sort of 20 years ago. And it's been looked at by various people over the years, particularly people such as Jose Carrello and Murakara have all kind of come up with sort of various sort of models to describe this process. And basically a kind of standard sort of model form for this sort of process is basically of this kind of non-local advection diffusion framework. So in this sort of model, the advective movement of the cells is driven by the adhesive interactions, which can be, of course, the kind of self-adhesion, that kind of red and red or U and U type cells, and the cross-adhesion interactions between the red and the blue, or between the U and the V cells. So that kind of comes into that non-local term. And a kind of a nice sort of recent addition has been to kind of have a kind of some sort of a population pressure term in there as well, that sort of is, generates sort of, is proven to be a kind of good way to, good phenomenon to include in this model. And these models basically are, kind of one of the key parameters in there, in addition to these adhesion strength parameters, is what we call the sampling range, which of course is sort of effectively modeling the fact that cells can only interact with other cells up to some maximum range, which will be roughly according to like the size of the size of their cell bodies plus any sort of protrusions that they may be extending from. So this is kind of the typical sort of model being used to describe this. And it proves to be actually very successful at describing this sort of encapsulation process, in that if you kind of start with a kind of a randomized mixture of those two cell types, then you see and set the parameters to kind of generate an encapsulation type scenario, then you do indeed see that sort of sorting process where those populations rearrange themselves, that you eventually get the blue population encapsulated by the red population. And even more encouragingly, it kind of is able to pretty well recapitulate all of those different sort of sorting patterns by changing the parameters of this sort of basic model. So you can kind of, if you choose the parameters appropriately, you get the right sort of mixing case, you get the encapsulation case and the various other scenarios. So that kind of suggested to us that this kind of non-local advection model is actually kind of a quite solid way to describe these kind of interaction-based movement terms that you get in cells. So I'm now going to kind of use the the next sort of 20 minutes or so of this sort of talk or so to talk about this phenomenon of chase and run. So what do I mean by chase and run? Well, chase and run is something that we kind of all know from playing in the playground ourselves as kids. It's something, and this is something that's kind of goes back over thousands of years. I mean, it's possibly to very, very early human populations. And there's a famous poem, the Distaff by Arina, who was a ancient Greek poetess, where part of that passage reads, "From white horses with madcap bound into the deep wave you leapt. I catch you, I shouted, my friend, and you, when you were tortoise, ran leaping through the yard of the great court." And she was effectively describing, recalling these childhood games of chase and run that she was playing. And of course, we continue to play this today. And I think in 2019, there was the kind of the official world record for the largest tag game, where I think 10,000 people assembled to play one of these kind of these games of chase and run. So chase and run, as I say, is something familiar to all of us from the playground. But it's actually a fairly ubiquitous phenomenon across the biological world. And it happens both in, not just between humans, it happens between predators and prey, but it also happens between a lot of different cell populations. And to show a couple of examples of this, one example, very much like a predator-prey type scenario, is this famous video of an immune cell chasing down a bacteria. And that kind of immune cell is kind of continuously kind of pursuing it. It's sort of detecting chemicals that is being produced by that bacteria, and eventually it manages to capture it and eliminate that bacteria. So chase and run is observed between immune cells and bacteria, and it also occurs between other cells, cell populations. And again, another very nice sort of video here is sort of the interactions between two types of fish pigmentation cells, xanthophores and melanophores. The xanthophore can be seen as this slightly kind of yellow blob here. The melanophore is this kind of larger, blacker blob cell here. And when these are placed on a petri dish, you can see a phenomenon of chase and run happening, where the melanophore is always moving away from the xanthophore, and the xanthopore is always pursuing it. So, and keeping this kind of interaction between them. So chase and run, as I say, we're familiar with it ourselves, but it also seems to be a process that is occurring down at the cellular level as well. So that's kind of what I mean by chase and run interactions. But now I'm going to just sort of like extend that concept to what I call collective chase and run. And my motivation for this is really kind of, again, a problem from developmental biology, and in particular, a process of neural crest migration. So for those of you that are aware of the neural crest, it's one of the kind of the early embryonic organs. And neural crest migration is a very fundamental developmental stage where certain cells, these neural crest cells, leave the site of their origin and then migrate, disperse across the embryo and contribute to various types of tissue in the developing embryo. And this is absolutely kind of fundamental for correct development, because if failed migration can lead to a variety of embryonic pathologies, which are called neurocristopathies. So the particular relevance of this to neural crest migration are these observations that were kind of recorded in about sort of 10 years or so ago by the Mayer lab, which took certain types of neural crest cells. So these are kind of cranial neural crest cells from Xenopus, and they placed these neural crest cells, this kind of big blob of green cells here, together with a population of what are called placoid cells, and this is a cell population that is adjacent to the neural crest during embryonic development. So this cluster of neural crest cells contains maybe 100 or so neural crest cells. The blob of red cells contains 100 or so placoid cells. And then when plated, the two populations then engage in a collective chase and run phenomenon. The population of neural crest cells pursues the population of placoid cells. And that's, in this sort of particular experiment, that's sort of continuing over sort of 5 hours or so. So this is, as I say, rather than kind of like an individual chase and run, this is what I would therefore describe as a collective chase and run, where rather than just between sort of two individuals, we're talking about kind of chase and run between two groups or two populations, and they're moving in this coordinated way, seemingly robustly for a considerable period of time. So the questions, therefore, from my perspective is what are the key processes and mechanisms that would allow this sustained and robust collective chase and run behavior? So that's kind of what we wanted to look at. And there's been quite a lot of experiments done on this, particularly by the, say, the Mayer group that kind of did these original, came up with these original observations. And the first one is that there's this process of contact inhibition of locomotion between the cells. So I kind of described that earlier, that kind of this idea of contact inhibition of locomotion describes the process where two cells move towards each other. They're on a path towards each other, they touch, and then they reverse polarity, and then they move in an opposite direction. And this happens, this is observed between all of these different cell types, between neural crest cells and other neural crest cells, between neural crest cells and placoid cells, etc. They all have this contact inhibition of locomotion. But then there's other things that are then acting on top of this. And one of them is that the placoid cells have strong self-adhesion. So they strongly express what's called E-cadherin. That's one of the kind of the principal cell-cell adhesion molecules, and that acts to keep the placoid cells together. So it kind of effectively overrides that contact inhibition of locomotion response. Then there's also a probably slightly more moderate mutual attraction that goes on between the neural crest cells. They release a ligand that seems to act as a kind of a chemoattractant that keeps the neural crest cells together. And that also seems to somewhat override that contact inhibition locomotion response between the neural crest cells. And then there's also a process where the placode cells attract the neural crest cells also via a secreted chemoattractant. And that seems to effectively override the contact inhibition locomotion response between the placode and the neural crest cells. So these kind of four phenomena together basically kind of give rise to the following set of interactions where neural crest cells are attracted to each other, placode cells are attracted to each other, placode cells are repelled by the neural crest cells through this contact inhibition of locomotion, and the neural crest cells are attracted by the placode cells. So there's your kind of chase and run there. The placode cells escape the neural crest cells. The neural crest cells are attracted by the placode cells. So to investigate this, we kind of consider two kind of populations of the chasers and the runners. These are, my chasers are the neural crest cells, the runners, the placode cells. You could take this model and put it into an ecological perspective if you kind of thought about your chasers as wolves and runners as bison, etc. But as well. And we kind of have the following set of interactions where you've got those, the kind of the chase, the self interaction that happens across some range, the kind of runner self-attraction, which occurs across some range, and then the chase and run, which again is going to be happening over some sort of range of interaction. So those are kind of the three kind of key elements that we include into the model. And then the question we asked is that if you kind of have this set of interactions and you kind of consider a mixture of chasers and runners, what do you get at the macroscopic level? And you could conceivably expect that you may be able to get this collective chase and run behavior going on, where those kind of they kind of sort into two separate groups and then move in persist in moving in this kind of collective chase and run. But you could equally imagine that they may just simply separate out from each other and kind of form into kind of two different sort of groups. So we wanted anyway to investigate what behaviors we would get. So we came up with a model for this, and this is work with Valeria Junta, Sada Bernardi, and Jonathan Potts, basically, that appeared last year in Interface. And it's basically just that same non-local advection diffusion model that we considered earlier, where you have the kind of the various interactions between self interactions and cross interactions between the populations. And in order to kind of set this up in a way to describe this chase and run behavior, the key things was to kind of set these various sort of interaction strength parameters, and to describe the self-attracting homotypic interactions, those kind of self interactions. We would set the two corresponding parameters to be positive, and to describe the chase and run between the two different populations, we set the alpha CR to be positive, but the alpha RC to be negative, to describe this kind of chase and run sort of behavior. So that was the kind of the model we came up with, and we wanted to explore its dynamic. And if you set the two populations next to each other to start with, kind of a population of red chasers and blue runners here, you put them adjacent to each other, you can actually generate nice, stable collective chase and run behavior. They forms what looks like a kind of a traveling wave type dynamic, where they just continuously move in a coordinated fashion while kind of maintaining a certain contact. So it does indeed generate collective chase and run, but not robustly, because then you change slightly some of these interaction strength parameters. And rather than seeing this collective, even though you're starting with the same initial conditions, for other kind of parameters, you get a kind of a segregation occurring where they just split away from each other and form two separate groups. In another case, the chasers may capture the runners and they then remain fixed. Or you even see sort of various instances of time periodic behavior, so kind of just continuous sort of oscillations behavior. So the model does generate collective chase and run, but not robustly. So then at that point, we really then wanted to understand what you could add to actually get this collective chase and run in a robust way. And that's when we thought to look at the ranges of interaction. Because so far, those kind of simulations that I've shown, we were assuming the same range of interaction for all of those processes. The distance over which the chasers were interacting with themselves was the same as the distance with which they were interacting with runners, was the same as the distance that runners were interacting with themselves, etc., etc. So we kind of chose those ranges of interaction to be the same everywhere. But it's probably not likely to be the case, because that kind of contact inhibition of locomotion, that's likely to be a short range response. It's mediated by cell-to-cell contacts. That's going to be a short range process. Similarly, that kind of cell-to-cell adhesion between the placoed cells is also likely to be a short range process. You need the cells to be physically touching at their membranes in order to do that. But then some of those other sort of processes in there are likely to be acting at a longer range because they're mediated by diffusible ligands that can spread into the space and initiate an interaction over a slightly longer range. So you've slightly got different potential ranges of interaction between them. So that then kind of that kind of motivated us then to kind of look at different possible configurations for the extent of the range of over which the chasers can interact compared to the range over which the runners could interact. And effectively assuming these kind of two scenarios where either the chasers have the larger range of interaction or the runners have the larger range of interaction. And effectively within the model, that all kind of then comes down to effectively describing what these various sort of xi parameters would be. We described a kind of a general chaser range and a general runner range. And now look at this effect on the patterning dynamics. So taking that kind of allows us then to kind of divide the parameter space into these two ranges into accordingly to where the chasers or the runners have the larger range of interaction. Those are kind of my two sort of regions of the parameter here. Run the simulations then and see what sort of behavior we get. And at that point, you get either robust chase and run or robust stationary patterns. When the chasers have the larger interaction range, you generate collective chase and run in a very robust manner. When the runners have the larger interaction range, you generate stationary patterns, very robust. And this does indeed seem to be an incredibly robust process in that if you change those interaction strength parameters, this is where my kind of chasers have the larger interaction range. Pretty well wherever I select, I get a robust collective chase and run behavior. So it would suggest those kind of different ranges of interaction could be crucial for generating these sort of this collective chase and run in a robust manner. But we also wanted then to kind of look at another things, and everything I've done so far has been just done a one-dimensional domain. And because that's also potentially going to be a very important determinant of pattern robustness. Because if you think of a chaser and runner in 1D, like kind of a wolf and a deer, in 1D, movement, of course, is massively restricted. The deer can only escape in one direction. It's not going to kind of go towards the wolf. It's going to run away from it. But in 2D, of course, you have numerous escape directions. And potentially, this could give rise to even more variety of patterning. So we wanted also to kind of investigate how robust this was going to be in the plane. And again, effectively looking at that kind of splitting it into interaction range, chaser and runner interaction range, we looked across the parameter space and to evaluate what sort of pattern we would obtain. And you get effectively the same fundamental behavior. You get in the region where before we saw stationary aggregate patterns, we still see stationary aggregate patterns. They can sometimes take slightly different sort of forms, but effectively there's no coordinated chase and run behavior in this sort of region. But in that region where the chasers have the larger interaction range, we continue to get an organization into a highly robust, sustained chase and run process. So this, again, seems to sort of reinforce that actually, if you have these, this particular set of interactions with the appropriate interaction ranges, you can generate highly coordinated collective chase and run behavior. But 2D does turn out to be a little bit less robust than 1D because it brought out a variety of what we call kind of exotic patterns as well. Because actually, if you then further explore this, and in particular, take an example here where the runners have a very small interaction ranges, you do see a kind of a chase and run process, but now in a quite sort of different sort of fashion where the runners basically form lots and lots of individual sort of groups. So they're not able to kind of stably stay together as all in one sort of coherent sort of group. They kind of maintain different sort of groups. And then the chasers are kind of course, moving around according to those sort of different sort of runner populations. So, it's a kind of a collective chase and run process, but not that kind of really highly organized sort of form where you get just sort of like a single group of chasers pursuing a single group of runners. You get kind of a much more kind of complicated dynamic going on in that instance. And further exploration of that kind of 2D sort of parameter space revealed a whole variety of different sort of patterns, ones where rather than kind of multiple groups of runners and a single group of chasers, you could get the reverse scenarios where you get kind of multiple groups of chasers and a single group of runners. You could get kind of highly regular sort of oscillating sort of dynamics going on. And you could also get kind of quite highly kind of incoherent sort of what we call incoherent chasing, where they kind of form some sort of chase and run dynamic, but not in a very kind of organized and seemingly almost sort of chaotic sort of fashion. So there was clearly a lot of sort of further behavior that could come out from this 2D model. And as I say, I don't know how well some of those movies came up could be viewed, but you can kind of view the movies here. If you kind of go to the paper, you'll be able to kind of see the various movies. Okay, so just to summarize then, and then kind of give a few words on where we're now kind of going with this, basically. What we've observed is that these kind of chase and run interactions can lead to a very broad variety of patterns when moved to this kind of macroscopic level. And particularly important for the robustness of these patterns are to have kind of distinct interaction ranges. So we in particular were looking at this kind of case where when the chasers have a larger interaction range than the runners, that could give a very robust collective chase and run behavior. And then in the context of those kind of placoed neural crest cells, what this model has allowed us to do is effectively identify a kind of a basic set of interactions and requirements that are capable of generating coordinated collective chase and run of the cellular group. So, and that's kind of helped to sort of feed back into the understanding of that system. Now, just to give you an indication of a couple of the next steps that we're doing in. So one of the kind of extensions that we're currently working on is to then look at this chase and run in the context of follower leader systems. So in that kind of in those kind of earlier simulations there, I just had chaser and runners, but there was no real overall kind of direction that would be taken. So there was no one particularly saying we have to go over to this particular sort of point. So another very kind of hot topic in the context of collective cell or collective animal movement is the idea of kind of followers and leaders, where you have a population that is effectively leading the rest of the group. And in the context of this particular model, the question that we're kind of currently working on is to add this sort of leader type dynamic into the system. And that could effectively be done in two different sort of ways. So you could assume that the chasers are leaders, and you could effectively model that very simply by giving the chasers some sort of advection towards some proposed target, and that would be your kind of chasers being leaders, or you could have the runners being the leaders and kind of have it giving them that sort of extra advection towards a specific target. And we're kind of currently investigating that model with the kind of the key question of seeing is it going to be more effective for one or the other population to be the leaders in order to get a particularly targeted sort of motion. So that's one of the things that we're kind of currently working on. And another one, of course, is, you know, is to kind of explore more into the analytical situation of this situation. And in particular, kind of a key question there is whether or not we actually have traveling wave dynamics in this system. So we seem to have a kind of a very robust, sustained chase and run process, but there's still the question of whether or not this is actually capable of generating true traveling waves, or whether or not this kind of chase and run behavior will eventually collapse. So we're kind of moving in either of these sort of directions, and various other ones as well. If anybody's kind of got any interest in any of those, please do feel free to contact me. So I'll just then kind of conclude then with a kind of a summary of where this work has been taken on. So I've been working on these kind of non-local advection diffusion models for quite a number of years with quite a lot of people, actually, that I don't have room to kind of thank all of the people that we've worked on, basically. But if anybody's kind of interested in these models, then a kind of a place to start would be this review article that I've written with Thomas Hillen and Jonathan Potts that was published last year. So, and that kind of gives a bit of background to that. This particular work on the chase and run behavior in heterogeneous populations, this has been worked with Valeria Junta, Jonathan Potts and Sarah Bernardi, and that appeared last year in the in Interface. So that's where we kind of described that. So I kind of leave it there, and thank you very much for your time.